everyone. So I'll be talking about Martian metabolism and biotic perchlorate reduction in Mars analog environments on Earth. So since the discovery of, let's see how I can get this to go. Since the discovery of perchlorate on Mars by the Phoenix lander in 2008, it's been a compound of interest on the red planet. This is due to its high oxidative potential on par with oxygen, which means that it's a potential energy source for life on the red planet. On Mars, it's ubiquitous in Martian regolith, reaching concentrations of 0.5 weight percentage. On Earth, there are microbes that reduce perchlorate in place of oxygen in metabolism, using it to drive the electron transport chain. And perchlorate accumulates in Mars analog systems on Earth, such as the Atacama Desert, where it reaches concentrations up to 0.1 weight percent, as well as the Antarctic dry valleys, where it reaches concentrations up to 0.003 weight percent. These are Mars analog systems that are incredibly dry and accumulate perchlorate via atmospheric deposition over long time scales. And they're likely, looking at these systems, it seems reasonable that perchlorate-reducing microbes might evolve and persist in these types of environments. However, the model organisms that we use to study biotic perchlorate reduction don't come from Mars analog environments. They come from the Potomac River in Washington, D.C., where there's very little to no natural perchlorate that accumulates. All of the perchlorate that we do find here is waste from munitions factories that gets dumped into the river. So how did perchlorate-reducing microbes arrive in this type of environment if they likely didn't evolve here due to the lack of perchlorate? Well, we know that the perchlorate reduction genomic island is widely spread via horizontal gene transfer. We know this because when we look at perchlorate-reducing organisms, can you see my mouse? Yeah, there we go. The elements required for biotic perchlorate reduction, the genes that are required for perchlorate reduction, share a lot of homology between the genes, but where the genes are situated in the genome is very diverse, depending on the organism that you're looking at. So it's a very chimeric metabolism. So if we look here, the bright green, that's chlorite dismutase, and this long dark green, that's perchlorate reductase. They're always kind of in different configurations in relation to each other in the genomic island. And that's because these individual elements are getting passed individually and independently of one another via horizontal gene transfer. So you only get perchlorate-reducing microbes when these elements come together in the same organism. So my research question was, did canonical biotic perchlorate reduction arise in these Mars analog environments like the Atacama Desert or Antarctic dry valleys, and then disperse around the world via acellular and microbial dispersal? So in order to test that, I selected metagenomes from sites around the world. A metagenome, for those of you who don't know, is basically all the DNA in a sample. It comes in little 20 base pair chunks, and you have to piece them together like a puzzle, and then pick out the puzzles that you're interested in, that maybe make a picture you're interested in, and then analyze those independent of the rest of the metagenome. So basically what I did is I selected metagenomes from sites around the world. I analyzed the presence of perchlorate reduction genes, so basically built the little puzzles and then pulled out the ones that made perchlorate reducing genes. And then I calculated the phylogenetic diversity of those genes at each site. This is a study that has really up until this point been done only in humans, which is this a type of analysis where we look at diversity of a species or a gene to determine the likely places where it's persisted the longest. That's how we know that humans come out of Africa, is because most of our phylogenetic diversity can be accounted for in Africa. So when I did this, so this is for chlorite dismutase, a gene that's involved in perchlorate reduction. I found the highest diversity to be in the Antarctic dry valleys. So what we're seeing here is these bars are a scale that's Faith's phylogenetic diversity. 0 is basically zero diversity, and then up to 50 would be the maximum amount of diversity in these genes that we see. And each of these blue dots is a separate metagenome. As you can see, there's a little bit of sampling bias because in the Antarctic dry valleys or Atacama Desert, we have a lot more sampling. There's a lot more research that happens in these sites. We've got a lot more metagenomes that I can analyze. Whereas if I look at other Mars analog sites like the Namib Desert, there are quite a few less, or in South Australian Desert, much less access to that kind of data. When I looked at perchlorate reductase, another gene involved in biotic perchlorate reduction, we also see that it reaches highest levels in the Antarctic dry valleys. So that basically indicates that these genes have persisted the longest in these Mars analog ecosystems, supporting the claim that these may have evolved and persisted in those Mars analog systems. So some conclusions: perchlorate reduction genes are present and diverse in Mars analog systems. That's really awesome, right? Before, we didn't even know if they could persist in Mars analog systems, but it looks like they both evolve and persist there for long time scales. This metabolism may have evolved in these environments and then prior to dispersing around the world via acellular and microbial dispersal. And studying perchlorate-reducing organisms in Mars analog environments can give us further insight into how life may persist on the red planet. When we're studying life forms that do metabolisms like what we expect to find on Mars, we shouldn't be looking at organisms that come out of the Potomac River. We should be looking at organisms in these Mars analog systems. So I'd like to hear some people I'd like to thank. So my funding, which is the NSF Grant for Long-Term Ecological Research at McMurdo Station, my department at Brigham Young University, as well as the help and advice of Dr. John Coates at UC Berkeley, Dr. Kendall Lynch at the Lunar and Planetary Institute, and Dr. Jennifer Glass at Georgia Tech. So thank you.